Hey everybody, Dr. Allis here. We are continuing our discussion of Chapter 3, which is all about cells. In this short lecture, we're going to go through and talk about the structures seen in the plasma membrane of the cell. When we talk about the plasma membrane of a cell, remember that the plasma membrane is my dividing line from outside the cell compared to inside the cell. Because the fluids on the inside and outside of a cell are different, we need a barrier to separate them from each other. The barrier that is the plasma membrane is made of two things. It's made of many phospholipids. So these are things that are a lipid layer with some phosphates attached. We also see several kinds of proteins, either inside or on either side of the plasma membrane. When we talk about the structure of the plasma membrane, we say that it follows what's called the fluid mosaic model. So a mosaic, like we see down here at the bottom, is something made of many different pieces. And the plasma membrane is made of many different pieces. But what makes this mosaic different from other mosaics is the fact that your plasma membrane is constantly changing. The proteins that we find embedded in it can move around. The lipids that are inside of it definitely move around. So it's a work of art that's made of many different pieces that are constantly moving around. When we talk about the plasma membrane, there are actually three kinds of lipids that build it. The first kind of lipid that builds the plasma membrane is called phospholipids. So phospholipids make up about 75% of all lipids in that membrane. They can have either saturated or unsaturated fatty acid tails. When we look at our picture of a phospholipid over here, we can see two tails on this phospholipid. The tail that lays reasonably straight, that's going to be a saturated fatty acid, whereas over here on the right, how this tail kind of kicks out, this tail would be an example of an unsaturated fatty acid. So if we have an unsaturated fatty acid, we can't squeeze those phospholipids together quite as well, leading to this little kicking out that we see of that tail. When we look at a phospholipid, the head group up here at the top is considered polar. When I say that something's polar, there are several things that should come to mind. The first thing that should come to mind is something that is polar would have polar covalent bonds. Polar covalent bonds are bonds where we share electrons, but we don't share them equally. Something that doesn't share their electrons equally is also something that has dipoles. So dipoles are areas of partial positive or partial negative charge. If you are polar, you have dipoles. But the last thing that polar means is that I am hydrophilic. Hydrophilic means water-loving. Things that are polar are water-loving things. They like to be around water because of water's dipoles. They get along well with each other. I spent so much time talking about the polar head groups because when we talk about the tails down here at the bottom, these are nonpolar. And when I say things are nonpolar, we can go through and make the same list of what that means. Nonpolar things have nonpolar covalent bonds, which means they don't have dipoles, which means they don't play nice with water. So we've got a molecule that half of it loves water, half of it hates water, and it's going to come together to form a plasma membrane for me. When we look at the plasma membrane, the nonpolar parts that don't like water are squeezed together, while the polar parts that do like water are either next to the extracellular fluid or the intracellular fluid. By forming this bilayer structure with two rows of phospholipids, we make all the parts of a phospholipid happy. Nobody who doesn't like water has to touch water. The water is squeezed to the other sides. The next kind of lipid that we see in the plasma membrane, making up about 20% of those lipids, is cholesterol. So we mentioned that cholesterol can help us make things like hormones, but cholesterol is also very important in the plasma membrane. That's because cholesterol has the ability to squeeze in between the lipid tails of the membrane. As it squeezes in between those tails, it does a couple of different things. The first thing it does is it helps to stabilize the membrane. By not leaving open spaces like we'd see down here where there's no cholesterol in this membrane, we're going to keep that membrane stable. We're going to keep it in about the correct location. At the same time, by having these little wedges inside the plasma membrane, that actually allows other phospholipids to move or to trade places inside the membrane. My final function of cholesterol in the membrane is it helps to keep things fluid. This is because the cholesterol can move just like those phospholipids can move. So cholesterol making up about 20% of the lipids in the plasma membrane, very important for stability, but also fluidity. The final kind of lipid that I find in the membrane is called a glycolipid, and glycolipids are the least common kind of lipid in the membrane. Only about 5% of the lipids in the membrane are these. When we talk about a glycolipid, its name tells me its structure. So glyco means sugar. Glycolipids are lipids that are attached to sugars. So if you notice in our picture, we see these areas where there's little purple sugars attached directly to some of my lipids in the membrane. Glycolipids are only found on the outside of a cell's membrane, and the job of glycolipids is to form a name tag for the cell. Your immune system will constantly scan these sugars that are on the outside of your cell to decide if you are or are not a normal cell. If you're not normal, I will do an immune reaction against you. If your name tag checks out, you're good to go. So far we've talked about 3 kinds of lipids in the membrane. We had the phospholipids, which make up most of the membrane. We had the cholesterols, which help to stabilize the membrane or allow a little bit of movement. And we had these glycolipids that form my name tag. Now we're going to switch gears and talk about the proteins that are in the membrane. There are many different kinds of proteins in the plasma membrane of a cell. Some of those proteins are called integral proteins. And when I talk about integral proteins, I could also call them a transmembrane protein. Transmembrane means that I go all the way from outside the membrane, all the way through it to the inside. So a transmembrane protein from the outside to the inside. Because I have this pathway from outside to inside, I can use integral proteins to do a lot of different things. For example, I can use them to move things from the outside to the inside, or vice versa. I can use them to send things from inside to the outside. So one function of integral proteins is transport. They help us to move things. Another function of integral proteins is to act as a receptor. So when I have a message that came from another cell outside of me, that can attach to a receptor, which allows that message to go into my cell and cause things to happen. The final function of my integral proteins is that some of them that go from outside all the way to inside are also enzymes. So enzymes, as we talked about in chapter 2, help us to perform chemical reactions and perform them quickly. By having some of these enzymes embedded in the membrane, we can do reactions very quickly. So integral proteins going all the way from the outside to the inside. We also have proteins called peripheral proteins. And peripheral proteins are proteins that are either attached just to the outside or just to the inside. These aren't embedded in the membrane. Peripheral proteins will attach to those ones that are embedded in the membrane, but they're not going to be part of what passes through. So peripheral proteins are things like the cytoskeleton, which, as its name suggests, is kind of like the skeleton for your cells. It gives them their shape. Peripheral proteins also form some of the proteins that we see outside a cell. Those proteins can perform functions out there. They could anchor other cells to each other. We find them outside. Peripheral proteins are also our motor proteins. So you've heard of actin or myosin, the proteins that slide past each other when a muscle contracts. That's an example of a peripheral protein. The last kind of protein that we have in the membrane are called glycoproteins. And glycoproteins, just like glycolipids, are proteins attached to sugars. Their function is the same. This is another name tag, but it's also a way for our cells to interact with each other. So by having a sugar on my surface, I can communicate with my neighbor. They can recognize me, or we can do a chemical reaction. With glycoproteins, we also see a very specific example, which is the AB blood typing system. So the AB blood typing system is based on which sugars you have attached to your proteins. So I'm not going to ask you specifically which one has galactose, which one has fructose. I don't need you to know that. But what I do need you to know is that the AB blood system is based on sugars that are attached to proteins that are embedded in the membrane. So glycoproteins and glycolipids both help us with cell-to-cell recognition. Glycoproteins also help cells to interact with each other. And when cells are interacting with each other, there are a few specific types of junctions or connection points that they often use. So let's talk about these connection points that attach our cells to each other. The first kind of attachment point are called tight junctions. And when we talk about tight junctions, these connect cells together tightly. So tight junctions are going to use a special kind of protein called an occludin, which occludes other things from getting between these cells. I would use a tight junction when I don't want anything to pass between my two cells. For example, stomach acid shouldn't be leaving the stomach. Therefore, I'd use tight junctions to attach the cells of the stomach to each other to make sure that the stomach acid stays in the stomach. So tight junctions, really good at making sure nothing can get between our cells. Sometimes it's not that I need nothing to get between my cells. Sometimes it's that I need my cells to be anchored together really tightly. And if that's the case, I'm going to use a kind of junction called desmosomes. When we talk about desmosomes, they have these really strong groups of proteins inside. They form this thing called a plaque. So a plaque is a group of proteins that helps us to attach these desmosome proteins to each other. They're anchored into the cell. We can get pulling in any kind of direction, but my thick cytoskeleton proteins, called intermediate filaments, they're anchored into the plaques and we can't pull those cells apart. So desmosomes, really good at resisting pulling. Tight junctions, really good at preventing stuff from getting through. The last kind of junction are the gap junctions. And gap junctions are where I literally have a gap or a space that I can send things through from one cell to another cell. So gap junctions are a type of junction that help me with communication, bringing my cells together. They have this gap or a tunnel that's inside the special kind of proteins that build these junctions. That's going to allow us to very quickly send a message from one side to another. So as you're considering these junctions, keep in mind that tight junctions say nothing's going between these cells. Desmosomes say nothing is going to pull these cells apart. Gap junctions say, let's talk. They allow messages to go from one cell to another. Different kinds of junctions with different jobs, but all of them connect the plasma membranes of neighboring cells to each other. So big picture, when we're thinking about the plasma membrane, what does it do? Number one, It's my physical barrier. It literally divides the inside from the outside. Number 2, as we're going to talk about more in another video, the plasma membrane is a chemical barrier, meaning it only lets certain chemicals in or out. Number 3, the plasma membrane forms my name tag by using things like glycolipids and glycoproteins. I put sugars on the outside of my cell to identify me, who I am. Number 4, using some of those integral proteins that go from the inside to the outside, I use the plasma membrane to receive messages, to hear what my neighbors are saying. And number 5, I use this membrane to make what's called the electrochemical gradient. Electro means electricity. Chemical means chemicals. As we'll talk about again later, the charge of the inside and the outside of the plasma membrane is different. And the chemistry, or I like to say the recipe, of the fluid inside and outside is also different. So we use the plasma membrane to do all of this to make things different inside and outside. You have an activity in your notes. We've listed the functions for you that the plasma membrane does. Go through using our discussion and using your notes and match up the names of these components of the plasma membrane with their function. Note, like it says at the bottom, they're not matched yet, but now that you've watched this video, I know you can get them matched. Good luck.